Attention, please. Right, while people are settling down, let me get going because we were way too long. Our speaker just told me that he has a 2-hour talk, so I think we should get going. Well, it's a pleasure to introduce Mr. Tom Mitchell. He is actually he has a very illustrious bio, and I read it and saw that it takes me about 5 minutes to read it, so I figured I'll cut it really short and stop after the first paragraph. He's the Fredkin Professor of Computer Science at Carnegie Mellon, the founding director of CMU's Center for Automated Learning and Discovery. He's the president of the AAAI, and most of the students in this room probably know him for his book titled Machine Learning. He's a member of the National Research Council, Computer Science and Communications Board, and I know him from something else. A couple of years ago, maybe 3 years ago, he took 2 years off to go and work in this company called BizBang Labs, which attracted some of the best researchers in speech and language. And apparently they had a great time for 2 years. So that's where I know about him from. And as you can guess, his area is machine learning. He's written a book on it. And I won't take any further. I'll just hand it over to him. And he's going to talk about something new that he's gotten interested in, applying machine learning techniques to brain function.
Thanks, Sanjay. Well, it is very, very nice to be here. And well, as Sanjay said, I'm going to talk about something a little different. When I returned to the university from my 2-year leave of absence, having a great time at this former company called WizBank, I thought it's time to figure out what I'm going to do when I grow up. And I also thought it was a good time to take a step back and reflect a little bit about the field of artificial intelligence and where it was and where the new trends were going to be coming from. The fact is, I think AI is in very healthy shape. And I'm not going to use my limited time here to try to convince you of that. I'd be happy to talk about it over a beer later with anybody who wants to get into the details of that. But one of the reasons that AI is in very healthy shape is that there's been a tremendously useful synergy between AI and another scientific field over the past decade, the field of statistics, that has really energized both of these disciplines, I think. You can see the influence of this synergy between AI and statistics in all kinds of areas, not least of which is natural language processing, where statistical language analysis is a major paradigm today compared to what it was 10 years ago. But you also see it in the field of machine learning, where statistical estimation and function approximation have become much more, the connection between those two problems has become much more apparent than it ever was. You also see it in areas like representation and reasoning in AI, and the use of Bayesian belief networks as a way of doing principled probabilistic inference, and also beginning to feed in some of the other ideas from AI about representation to extend those ideas. So why am I saying this? Because I think one way to understand where fields are and where they're going is in terms of the synergies that you get with other disciplines that happen to be aligned in some way with their goals. And over the past decade, if you ask me, I would have to say that it's been the field of statistics that has generated the most synergy, cross-disciplinary synergy with AI. If you ask me, what is the discipline over the next decade that's going to have the strongest impact on AI and vice versa? I actually think it's the brain sciences. And when I say brain sciences, I mean all of those fields that have to do with understanding how brains work, ranging from psychology to cognitive science to neurophysiology. And that's really the thesis of this talk. I never forgot what my thesis advisor told me, which is always state your thesis. So here it is. I really believe this statement, and the rest of the talk is really designed to try to convince you that you should believe it too, that the synergy between AI and those sciences that we'll lump together as the brain sciences is going to yield very profound advances in understanding of intelligence over the coming decade, and will really change the way that we think of AI in a way comparable, at least to the way that statistics has changed the way that we think about natural language processing and machine learning over the past decade. So why would somebody believe something like this? Really, I have three reasons. One is, it's obvious if you're looking for fields that are aligned with AI, the main alignment is we're talking about fields that have the common goal of understanding intelligent processes. That's what brain sciences are also about. Second, it turns out I was surprised when I started looking into it to find that there were many more and more remarkable correspondences between AI algorithms that were developed for good engineering purposes and what we're starting to understand about how humans and other animals process information. So there are, in fact, a number of significant correspondences already between techniques for things like robot navigation that people have developed for engineering purposes to control robots and what we're starting to understand about animal navigation. And in the talk, I want to sample a couple of these. The third reason, and the one that actually has me most excited, is that there's a revolution going on in the brain sciences. And it's pretty much engendered by the fact that there's new instrumentation. And often that's what causes revolutions in sciences, at least in the natural sciences. And through a number of new techniques for brain imaging and direct cell recordings, new dyes, genetic engineering, there are a number of different techniques for doing much more controlled and much higher observability experiments than ever before in the brain sciences. So these are the three reasons why I'm anticipating a very exciting decade for the synergy between these two areas. So in fact, the outline of the talk is to sample a couple of examples where I think there are already interesting links between ideas that have been developed in AI and what we're starting to understand about animals. And then also I want to talk about some of this new instrumentation, and in particular functional magnetic resonance imaging. And that's kind of the outline of the talk. So let's dive in. The first area where I think they're quite interesting, you can think of this as a case study supporting my argument that there are already interesting things that we can learn back and forth between AI and brain sciences if we focus in, for example, on the question of spatial localization. So imagine you have a robot roaming around in the hall, and one of the very basic things it has to do is figure out where it is. Well, people in mobile robotics have worked very hard on this problem over the past decade. They won't agree with this because they want to publish more papers, but it's pretty much a solved problem. It's pretty close to a solved problem. It's in the 80-20 scheme of things. It's 80% of the way to being a solved problem. So you can put a robot in this room and it'll roam around autonomously and build a pretty good map of the room and also be able simultaneously to figure out how to locate itself. What are the engineering ideas that they've come up with to make that a pretty much solved problem? There are just a couple. The first idea is that if you want to build a program that lets a robot figure out where it is in the room based on things like sonar and laser range sensors, idea number one is you don't want to have the robot represent its location as a position in the room. Instead, you want to have the robot represent its location as a probability distribution over positions in the room. Okay, so that's engineering idea number one that just turns out to work better than not using probability distributions. And then you can use Bayesian updates as the robot roams around the room to update that probability distribution on its location. So for example, if you imagine that we have a robot in a very simple hallway where let's say it's only interested in one dimension, which is how far down the hallway is it, then we might, when we turn the robot on, initially give it a uniform probability distribution over the hallway. You know, it doesn't know where it is, so it's equally probable that it would be at any x position given its model of the world. Then if it takes some observation, maybe looking with its sonars and laser scanners, that observation might reveal that there's some door-like indentation near it. Given that observation, it would update that probability distribution, giving it this posterior that would show that, well, it's more probable that the robot is in one of these three locations. You and I can see that's because that's where the doors are. Furthermore, if it moves forward, it could use its probabilistic knowledge about next state to update that probability distribution further, after which if it takes yet another observation, it can get more pinpoint estimates of where it really is. Okay, so in pictures you can see there the probability distributions being updated. This typical use of Bayes' rule. And that's idea number one, that the people engineering solutions for robotics came up with. Now there's a problem with idea number one, which is primarily that we don't really know what functional form this probability distribution will be in. And so idea number two, and there are only two, is we better represent this probability distribution not as a enclosed form, as a formula, but instead with a sample of points. So we use Monte Carlo methods and represent the distribution as a sample of points. So for example, in this more complicated map, when we turn the robot on and it doesn't know where it is, secretly I'll tell you where it is. It's here. And it's about to follow the trajectory given by these blue lines when I turn on the video. But when we turn it on and it doesn't know where it is, we represent that uniform probability distribution now with a sample of points uniformly distributed. And it can do the corresponding Bayesian update using Monte Carlo methods on these points. So as the robot starts moving around, notice when it turned on, it thinks the most likely position that it's in is actually where this green dot is. That's just because the dots happen to randomly be slightly more dense there than they are anywhere else. But you'll see as it follows this blue arrow trajectory and updates the position, I'll keep my finger on where it really is, sooner or later the green dot snaps to the actual position of the robot. Now you'll see it turn and go toward this door. You see there's still some probability associated with other aliased locations in the map, but eventually the robot collects enough data to disambiguate these things, and the illusory possibility that was over here kind of disappears eventually. Okay, so that's all I'm going to say about engineering of robot solutions for localization. Two key ideas: probabilistic representation of where you are, and represent that through a sample of points, represent the distribution using a sample of points. Okay, stop there. Okay, now, what do we know about localization and navigation in, say, rats? Suppose we put a rat in a maze, and we're interested in how rats represent their location in the world and how they update it. What do we know about that? Well, it turns out this is a problem that people have studied. One way they've studied is by putting implants in the brain of a rat and doing direct neural recordings from neurons in various parts of the rat brain, including the hippocampus. And interestingly, what they found is in the rat's hippocampus, there are neurons whose firing rate is very sensitive to the exact position of the rat within the maze as it moves around the maze. So what you see here is a simple maze that the rat is in. By the way, the north room and the south room visually are identical, so it's hard for the rat to know when you plop it in the maze whether you've plopped it into the north room or the south room. But anyway, as it roams around, if you record from one cell and put a dot whenever that cell fires, and also track where the rat really is, then you get this kind of a map that shows this particular cell fires when the rat's in this set of locations within the maze. Similarly, there's a second cell with a different firing map and so forth. These things are called place cells for obvious reasons. They fire based on the place where you are. Okay, so this is kind of interesting because it looks a little bit like the picture we just saw with the robot. It leads to the obvious question, are rats representing their location in the room in some way by encoding a probability distribution over locations in terms of these spiking rates of these particular collection of neurons? So how would you even answer that kind of a question? So one way you could try to answer that question is to say, well, can we think of a probability, can we make up a probability distribution that we define as observers of the rat, but that we define in terms of the observed spiking rates of these cells, and that has the property that the maximum likelihood estimate of where you are, according to that distribution, really is where the rat is. Okay, so here is the probability distribution that Skaggs and McNaughton used. Essentially, it's just the product of a term generated by each of these cells, where the term generated by each cell is just the firing rate of that cell. It's just the probability that, so the distribution over position x at time t is just the product of probabilities generated by each of these things. And each of those individuals are a probability distribution defined in terms of the density of dots in that particular location x and the spiking rate that we observe at time t. And so if you're seeing a high spiking rate, then you're going to assign more probability to the place where those dots are more dense. So this is one of many ways we could define such a probability distribution over positions. How do we know if this makes any sense? Well, what we should do now is we should let the rat roam in the maze, keep track of where it is, and also compute the maximum likelihood estimate of where it is according to this probability distribution that we defined in terms of those neurons. So they did that. Here's the answer. These, the solid line here shows the vertical, the y coordinate of the rat's position over time as it roams around through the maze. So this is where the rat truly is. The dots here show the maximum likelihood estimate of where the rat is according to that probability distribution we just defined. And there are a couple things to notice. First, here at the beginning part, it's wrong, but it just thinks it's in the north room, whereas it's actually in the south room. But after this point, it's pretty much right. That is, the probability distribution that we made up based on the observed spiking rates and densities for those place cells actually gives us a maximum likelihood estimate of the rat position that corresponds with the rat position, at least the y coordinate. And now I'll tell you the reason, what's going on in this part. This was shown, this data was collected during the final run of the rat through the maze. And every time that the rat was put in the maze in previous trials, they plopped it down in the north room and let the rat roam around. On the final trial, they plopped it down instead in the south room, which you'll recall is visually identical. If this probability distribution is to be believed, that we defined as the belief, defining the belief state of the rat, then what happened is the rat incorrectly believed it was in the north room and believed that it was roaming vertically up and down through the north room when in fact it was roaming up and down through the south room until it came out into the hallway and went up there and saw there was a north room, and then it locked into reality. So this is actually, if we interpret it that way, number one, evidence that we can make up a probability distribution over locations.
So is the probability distribution learned here?
Is it large?
Yeah. Is the learned here probability distribution? Is learned?
Learned.
Yeah, it's not learned. We simply define it this way. This is one of many distributions you can make up. And we simply define it this way.
Okay.
And the density here is the empirically observed density of the firing rate for that cell.
So parameters are not learned here.
No.
Parameters of Poisson distribution are learned here. No.
I see what you're saying.
You are considering Poisson distribution?
Yeah. The density and the spiking rate are given. If there are other parameters, I don't know.
So lambda parameters in Poisson distribution.
So I don't know.
Okay.
I'd have to go. We should go back and look at the paper to answer that. I see what you're asking now. So it's possible that those were fit. Okay, but the point remains that here's some evidence that can be interpreted in the following way. That stochastic firing rate of this collection of cells can be interpreted as encoding a probability distribution over locations in the maze. And in fact, the maximum likelihood estimate, according to that distribution, tracks pretty well where the rat really is. And if we believe that, it even tells us that we should think that the rat was temporarily confused about which room it was in, and it all kind of hangs together. Yes?
Just point of information. If I understand right, what you demonstrated is that in this vector of 7 firing rates, there's enough information to reconstruct the position. So that if the rat happens to have these 7 cells with these responses, then that's enough for it to, that's correlated with position, right? That we can reconstruct position.
We can reconstruct position from that. And in fact, one way we can do that is by defining that distribution.
Is by exactly this model. So the question is, these particular place cells, have they adapted to this particular maze? Or are they essentially fixed and we're just building something on top of them?
Yeah, so I think the truth is that the full answer to that question is not known. There are several follow-on papers where people are trying to understand how plastic are the definitions of these place cells. And obviously, the first time you go into the maze, what would it even mean?
Well, you want to know whether the response changes over time, right? So you let it wander and then you compare the first half of the trace to the second half or something.
So I've seen a couple follow-on papers that I've read, and they did not answer that question for me. But they did show that there was some plasticity in those things. All right. So anyway, I thought this was such a cool result. I had to tell you about it. Besides, it supports my thesis. That there already are some, here's one example of where people who didn't really care how rats work, but really wanted to engineer working robots, came up with a solution to how to represent uncertain information about location in the world, integrate sensory data, and get reliable estimates of robot location. Came up with a method that gives us one very useful way of interpreting what we're seeing when we start watching spiking rates of neurons in the rat hippocampus. So does that mean that rats work like robots? Well, not necessarily, but in fact, if we draw out the correspondence here, to kind of summarize, robots have these engineering solutions to have given robots these probabilistic representations using Bayesian updates, Monte Carlo methods, and they give us a joint distribution over the XY position plus the orientation of the robot. If you look in rats, we saw a similar story. We also see some differences. It turns out, for example, in rats, the head position, the orientation of the head is apparently controlled by a very different different set of neurons, encoded by a different set of neurons, than the XY position. So we also see when we start looking at the correspondence that there are some differences, which maybe is interesting to AI because it raises questions like, well, is there a good reason that evolution decided to encode this in a distinct subsystem, maybe there's some advantages. So I don't want to make this into a talk on spatial reasoning, but that's example number one of what I mean when I say there are already some interesting correspondences. Example number two. Close to my heart is the topic of machine learning and reinforcement learning as one variant of that that has to do with training systems to learn control strategies. So for example, if we had a robot roam around in a very simple grid world where it received a reward of 100 points whenever it entered this state in the grid, the goal state, one way for it to learn a control strategy is essentially to learn an evaluation function over states that might, for example, define the value of a particular state as the expected discounted future rewards that we would receive if we were to take the optimal control action from that state. If you didn't get all the details of that, that's all right. The important thing is, in reinforcement temporal difference learning, what we're doing is learning an evaluation function over states that has particular definition, and which once learned, if it's learned perfectly, gives you optimal control. This little grid world is a nice way to describe it, but to tell you how well it works, it's nicer to tell you that Andrew Ng just finished a PhD thesis at Berkeley where he used essentially these techniques to train a system to control autonomous helicopter, and has really the world's best control system for doing acrobatic maneuvers in autonomous helicopters now. So these are very real, well, again, this is an example of an engineering solution to certain kinds of highly nonlinear control problems. Why am I telling this? Because, as you may suspect, there's a correspondence to what we now are starting to see if we ask, what about animals? And if we give them rewards and look at how they learn, what do we see inside? So one way of studying that is to look at dopamine. Dopamine is a chemical in the brain that seems to be highly related to reward and learning to anticipate reward. And in fact, that's what this little diagram shows here. This diagram shows you data collected from a monkey, from individual neurons in a monkey when it was given, this is an axis is time. Each of these lines down here horizontally shows the spiking pattern for a particular neuron. And what you see up here is essentially a histogram of these spikes. And so what you see is over time, when a reward was given to the monkey, in this case a little sugar water on the monkey's tongue, what you see is an increased activity in the dopamine-related firing. So, in fact, people have noticed this in many experiments and generally now believe that dopamine is somehow related to a reward signal in the monkey and other animals. Apparently almost all animals. What's interesting is, thinking back to our reinforcement learning thing, is, well, what if we give a flash of light to the monkey a little bit before we provide the reward? The flash of light we might call a conditioned stimulus. Then after a while the monkey learns to anticipate the reward when it sees the flash of light. And what's interesting is if you record from those same neurons, you see the dopamine-related spiking occurs when just after the light is flashed. And when the reward is given, actually we don't even see it. Which is actually the same behavior that you would receive, which you would see if you were looking at the variable in your reinforcement learning system that is calculating the difference between the expected reward you'll receive in the next state and the actual observed reward. So here there's no surprise, no blip, because your expectation matched reality. Here there was a surprise because suddenly you went from no light to seeing the light, and now you expect high reward in the near future. Well, that's consistent with what we're seeing here. So it may turn out, in fact, that this dopamine signal is somehow doing a back propagation of reward signals to earlier anticipated state. There are many details left out here, but there's, again, an interesting correspondence. And just to finish this story, here's the kind of a interesting third experiment, same setup, but now what if we flash the light and don't bother to provide the sugar water at the end? When we flash the light, we still get, of course, the anticipating dopamine spike. But now when the sugar water doesn't come, we actually get a drop in the dopamine-related stuff below baseline, which would be what we'd expect that value of difference between anticipated and observed reward again in the reinforcement learning scheme. So this is very interesting evidence of some kind of relationship between the kind of reward differences. These things are called Bellman updates. They even have names in the reinforcement learning literature, between those particular quantities that are key to doing reinforcement learning in our computer systems and the spiking of these dopamine cells and systems. So there's a second example where, again, we have AI solution developed, and now we start to look at what's going on in animals. And it doesn't tell us what's going on in animals, but it gives us a very useful framework for at least making conjectures about what might be the learning mechanism here and seeing if the data that we're observing is consistent. And in this case, in fact, it is. Yes?
On the flash of light, was there any other kinds of temperatures, right?
Say that again?
On the flash of light, was there ways or kinds of CS that were tried? Apart from providing the flash of light, then there was there anything else that was tried? Like, say,
Oh, no.
color
Yeah.
tone or something like that?
Um,
Or was it
I don't know. I don't know. So what I remember from reading this paper was just the flash of light. There have been many, many other, again, several follow-on papers on this since then. I just don't know. Okay, so I could tell more stories like this. I'm not going to. I'll just tell you that, just like before, once we start looking at how the rat does navigation and getting data, it kind of made us ask questions about our current AI solutions. Similarly here, if you look at theories about animal learning, many people believe that there are actually quite different neural level things going on when animals learn from positive versus negative rewards, sort of sugar water versus very bitter day-old coffee. I'm not sure they did that experiment. Which is, and it's actually, I think more people believe that they're different, that they're handled differently in monkeys than believe that they're handled the same. Now, in fact, if you look at the reinforcement learning methods that we've developed for controlling helicopters, they treat positive and negative rewards as just the same thing, just with a sign difference. And if, in fact, it turns out to be true that they're handled differently in biological systems, that again leads to an interesting question. Well, why? Is evolution just dumber than we are? Or maybe not? So, again, we get some interesting questions from the crosstalk. All right, I could tell more stories. There's a nice one to tell about what we know about computer vision and animal vision, including some very interesting work by Tommy Poggio at MIT, looking at direct cell recordings from visual system of monkeys while they're learning to recognize objects, and how some of the cells in those monkey visual cortex have responses remarkably similar to the hidden layer in a radial basis function neural network. But I don't want to take my time to do that because I'd rather talk about brain imaging at this point. But I just wanted to give you a sample of several places where already you can see that we're starting to learn more about animals and how they do things. And the main theme of these three examples has really been that earlier work in AI gives us a kind of conceptual structure for trying to interpret these neural signals that we're seeing in animals. A very useful conceptual structure. Okay, but onto the, remember the outline was to show us some examples of that, and then to look forward at some of these brain imaging techniques. In fact, there are several kinds of instrumentation that I mentioned, as I mentioned already. But what I want to focus in on for the rest of the talk is one technique that many people are now exploring called functional magnetic resonance imaging, or fMRI. What it is for the first time in history, a way to non-invasively get high-resolution pictures in 3 dimensions of activity in your brain. In particular, you sit or lie in a big magnet, a big DC magnet, and then you think, or you don't think. In either case, it takes a snapshot in 3 dimensions of an indicator of brain activity. Actually, what it's measuring is fluctuations in your blood oxygen. And the idea is if this neuron up here starts firing intensely, it consumes energy, and that causes fluctuations in my blood oxygen in the same or nearby, very close by location. So we're not looking at neural firings directly, but we're looking at something associated with that. But what's interesting for our purposes is that it gives us these 3-dimensional images with a 1 millimeter spatial resolution. Roughly 30,000 3-dimensional pixels or voxels at a millimeter on a side. And we can get an image like that about once a second. So this is pretty interesting, right? If I told you I could give you access to one of these machines, you could start dreaming up some pretty interesting experiments to run on people. You know, what does their brain look like when they're thinking of their mother? What does their brain look like when they're thinking of their dog? What does the brain look like when they're hungry? What does the brain, you know? You could make up a lot of experiments. That's exactly what psychologists have been doing for the past 5 or 6 years. And there are just some really fun things that we now can see about the brain that we never could before. Here's a typical study. In this study, which is done by Marcel Just, a psychologist at CMU with whom I'm collaborating. This is some work that Marcel did with his group. He said, well, we'll give people mental rotation problems. And so the idea is put you in one of these machines, and I say to you, I don't actually show you a picture, I just, on the screen it prints in text. Imagine an alarm clock with the hands at 6 p.m. Now, rotate this in your mind 90 degrees clockwise about the vertical axis. Right. Now rotate it 90 degrees counterclockwise about the horizontal axis. Okay, now I show you a picture and I say, is this what it looks like? And you have to answer yes or no. All right. Now, you maybe get it right, maybe you get it wrong, but the point is now we've got captured a video of the brain activity while you're doing mental rotation. In fact, if you look at that, what you find is these yellow regions and red regions and blue regions are the parts of your brain that are much more active when you're doing mental rotation than when you're staring at just a blank screen or a screen with a little static dot in the middle to make and told to relax. So essentially, this is a prototypical case study for fMRI, use of fMRI to try to understand what parts of the brain are related to what kinds of cognitive tasks. And there are many such studies. I just want to show you 2 others that are 2 of my favorites in some ways. Here's one where people were not given mental rotation, but they're told, here's a word, and a word being presented every 5 seconds. Please remember these words. Because after you get out of the fMRI machine, 2 hours later, we're going to give you a test. We'll show you words, and you have to check off the ones that you saw when you were in the machine. So you got to remember these words. It turns out there's a spot right here in every subject's brain that they looked at, and there were roughly on the order of a dozen subjects, right up here, that's very active when you're trying to remember those words. more interestingly, if you wait the 2 hours and give the person the test and see which words they in fact did and did not remember, what you see is that the activity from stimulus onset over time, so here's time 0 is where the word's presented to you, and now you're trying to remember it. The purple line shows the activity in that location if I average over the words that were actually remembered versus in green the ones that were not. So that, if you believe it, that's a curve that's showing difference in the magnitude of activity in that spot that's predictive of whether you're going to remember that word 2 hours later.
What you're showing here is the mean activation in both cases. So I'm wondering about the variance and to what degree this difference could be used to discriminate the two conditions.
They don't report
Could I put this up to my class, right, and tell them when they should pay attention because this area isn't lit up, or is there too much error?
I'll tell you, there's an incredible amount of noise in these things. So these are average curves for a number of words. But, so what you're seeing is a difference in the mean activity among words that you remember and don't, which is not to say that for an individual presentation of a word, we can make the prediction. It might be too noisy. In fact, I will have more to say about that in a second, because that's some work that we've been doing. Okay, so but before we do that, I just got to show you, there are many things that people have studied with this, including this one. All right, so there's a sample of the kind of things that people have been doing, and it has shed a lot of light over the past 5 years on what parts of the brain are more active when you're doing task X. Now let's come back to AI and cognitive science and what we really are interested in.
Is true?
I'm just summarizing what's in the literature. So now, as AI people, we're interested in many questions. But, you know, the thing that gets me excited about fMRI and about brain imaging more generally and all these techniques is, of course, what we really like is to understand what are cognitive processes really doing in there. You know, we'd like to have models that talk about going through different states and transitions among those, and we'd like to have models that ground those out in observed fMRI activity over time. So, you know, if the machine allows sufficient signal to noise and sufficient resolution, maybe we could start looking at developing real models grounded in observed, directly observed data from human brains. Now, there are a couple things that make this difficult. One is that wonderful 1 millimeter spatial resolution that I talked about a moment ago contains about 10 to the 4th neurons. So it's still pretty fat compared to a neuron. The second is that the temporal resolution of fMRI is pretty slow compared to neurons. I mentioned we could take a snapshot once per image. Sometimes we do it, or once per second. Sometimes we do it twice per second. But the truth is, the blood oxygen fluctuations that we're measuring smear out over several seconds. So there's some temporal smearing going on. So we don't get good enough spatial or temporal resolution to look at individual neurons or individual firings of them. Nevertheless, it is a technology that looks at the whole brain. So maybe we can still develop models that do ground out in reliable temporal spatial patterns of fMRI activity and do this. So there's the challenge, and that's what I want to talk about the rest of the talk is this feasible at all? Is there any hope? Should we go back to some other problem, or should we get working on this? So first I want to present two pieces of work that I find very interesting in this regard. One of them is by a guy named John Anderson, also a psychologist and cognitive scientist, who has developed a cognitive architecture called ACT-R that he has used to model many human cognitive functions. And when I say model them, I mean things like, as people do arithmetic problems, including kids, and measures over time the errors that they make on different arithmetic problems and the response times. And for many years, John has been developing this rule-based cognitive model that tries to fit the observed data from people. And he's done a fairly good job of
But
it.
is it only the rule-based? There is no utility of case-based reasoning here. Actually, in some this process of learning, this process of
In the ACT-R model, it's done with rules. And John's answer to your question, if he was here, would be, I can put cases into rules. But it's all rules in this architecture. I'm not saying this is the right architecture, but here's what's interesting about it. In the last couple years, what John has done with this architecture, he said, well, why don't we use not just behavioral data like response time and error rates, but let's use fMRI. Let's have people solve algebra problems while they're in an fMRI machine and observe that data and further tune these models. So what he's done is taken, here's a block diagram of his architecture, which has blocks that do things like visual input, manual motor control, matching of the production rules. And what he has done is he's conjectured particular regions of the brain that he claims are responsible for implementing these different components. So for example, production rule firing involves 3 sections of the brain that in fact do 3 different steps: the matching of the rule, deciding which of the matched rules to fire, and then actually executing it. So he's making a conjecture about how the operations in his architecture correspond to distinct regions, cortical regions. Okay, now that he's done that, he's then gone the next step of collecting fMRI data from people solving algebra problems. In his experiments, he first presents some variables on a screen, then the screen goes blank 2 seconds later, and up comes an equation like this. Then the person, 1.5 seconds is the time scan. Now you're supposed to answer this, solve for x, and type in the answer on a little button. Then the screen goes blank and you get to wait for another problem. So that's the stimulus. You're sitting in the fMRI machine solving these algebra problems and doing it. Okay, now he wants to fit his model to that data. So what he does is he develops a production rule system that can solve algebra problems. And every time the architecture executes a production, those different pieces of the model, get involved and create some conjectured activity in some cortical region. So if we give his production system an algebra problem, what we're seeing here is the trace of steps done on a production by production basis. You can see, for example, it's remembering that C had the value 24. It's subtracting the 3, deciding to use subtraction to get rid of that 3 on that side of the equal sign, doing the subtraction, and so forth. So you see the kind of grain size of steps. And now, each time the system executes and does one of these steps, it uses certain parts of that architecture, which would correspond to a prediction of fMRI activity in those locations. So are those predictions right? Here's a plot that shows in the solid lines are the predictions for just one of those regions, the image buffer. The solid lines are the predictions of activity in the image buffer and the corresponding posterior parietal lobe of the brain by the ACT-R system as it solves that algebra problem. And the 3 colors correspond to problems of increasing 3 different classes of difficulty in the algebra problem. What you see in the dots are the observed fMRI activity in that location in the brain for the 3 different kinds of problems. So here is a very interesting result, if you ask me. It's showing that this architecture and these commitments about what parts of the brain are doing which steps in the architecture And then executing that architecture on individual algebra problems gives qualitatively good correspondence to what you see if you have people in an fMRI machine doing mental algebra.
I don't understand the seconds. What is the, this is the time it takes? It must be normal.
This is time. So you're presented with the algebra problem, and now you're solving it.
And we solve it in the same time regardless how hard it is?
Well, it's a little bit longer, but not too much. A few seconds longer. A few seconds longer.
But
And this
but
might-
it looks like it's 10 seconds after presentation.
It is.
Thinking, regardless of how hard it is.
So one of the unfortunate properties of fMRI is that the fMRI response peaks about 5 seconds after your neurons fire. So that blood oxygen fluctuation is a delayed and smeared out temporally thing. So that's part of why this is as late as it is.
Then architecture and several modules, is this just for one of the modules or which?
Yes. So let's see, we were just looking at this one, which corresponds to the visual buffer,
Okay,
and which he claims corresponds to the posterior parietal lobe. Here are similar results for two other regions that correspond to two other parts of his architecture. And these were the only three that they actually recorded from and tested on. But the point is not whether John's got the right model or the wrong model. The point is it's not crazy to start this kind of research where we start building procedural cognitive models and try to fit them to observed fMRI data. And I think this work is the first of what hopefully will be a much more popular research paradigm for cognitive science. Since I'm in the land of language processing, I can't resist telling you that if you want the 2-hour version of the talk, then I would have told you about Marcel Just's work on building a similar kind of architecture that's just looking at language processing. Instead, I'll just say that just like John, he has an architecture that makes predictions about activity over time in different regions of the brain while it reads sentences. And that just like John, he has some plots that show the model predictions and the observed fMRI data in these different regions are in some kind of correspondence. But if I did that, I wouldn't get to tell you what I'm doing. So I'm not going to do that. Instead, no, you can't read that yet. This is good. This is good practice to distill this into 3 minutes. As a machine learning person, interested in this new device, one of the most obvious questions is, well, given these complex temporal spatial movies of brain activity that we're getting, given that nobody really knows how to interpret these things, would it be possible to train a machine learning system to decode the mental state that a person is in by looking at these brain movies? If so, for what kind of mental states could we successfully do that? How much data would we need? Is this even possible? So those are questions that my group and I started looking at beginning of the year, and we've since then done a number of experiments to take some fMRI data and try to train classifiers. So I'll tell you about 2 of those. One is some work done with Marcel Just, where people were shown words that fell into one of these categories. And we were just interested in the question of whether we could train a system to detect whether the word you were currently looking at was a word about food or a word about people, based on the fMRI activity. So we set that up as a standard machine learning problem, where we're the function approximation problem, where the function we're trying to learn is the mapping from the snapshot at time t of observed activity to the category of word that you're looking at time t. Actually, it was t minus 5 seconds because of that temporal blur. Did this with a single subject, used only certain regions of the brain, but still thousands of voxels, and tried a couple different classifier methods, but the one that worked as well as any was Gaussian naive Bayes classifier, where essentially we use the training data. Now, the training data, of course, is here's the brain image, and we know the person was looking at the word fish. And here's an image where they were looking at the word house. So we're trying, then we use that training data to learn the probability distribution over fMRI images given the word category. And in a Gaussian naive Bayes classifier, what that means is we assume the activity at every voxel in the image is independent and can be modeled as a Gaussian distribution, one Gaussian for the for words in category I and a different Gaussian for words in category J and so forth. So having done that, then we asked, well, having trained the system, then we found, in fact, that the system could discriminate pairs of categories of these words with these kinds of accuracies that you see here. So, for example, it was possible for the system to tell whether, if we just show cases where you're known to be looking at either an animal word or a nature word, then it could get that with 83, it could predict that with 83% accuracy, whereas random guessing would get you 50%. And there's enough data that these things are very improbable very unlikely to have happened by chance. So it does look not perfectly, but to some degree, it is possible for the system to be trained to distinguish certain kinds of categories. We don't know why artifacts are commonly involved, at least in this experimental data, in hard-to-learn distinctions, but that's the experimental data. Now, you might ask, well, what do those learned models look like? Here's one. So here's the learned probability distribution for brain activity. These are the means that we're looking at, the mean of the Gaussian distribution for different voxels. Given that you're looking at, or given that this person was looking at a word in the category people. So you see they're kind of messy. There's some continuity. Red here means, the redder or the oranger means higher, and the darker blue means lower. So you see there's some sort of spatial continuity there, blue regions. It's not just a random dot pattern. It's kind of hard to know what to say about this picture, but I'll show you this picture. This is one, oh, I'm sorry. This is one person, and what you're seeing is the single three-dimensional image by slicing the brain horizontally. So there's one person, one model.
So you're learning the mean and the variance
for each
category
voxel.
and each box?
Yes. And then using those to make predictions about the next. So if you line up two of these next to each other, so if I show you this for the people category, you go, okay. It would be interesting to see, like, let's take one of these slices, this one, and let's put it next to the means that we have for some other category to see how they look. And so here's the one for animals next to the one for people. So, you know, there are quite a few similarities. There are some differences, especially down in this region, which seems to often, in many of these patterns, this region down here seems to have different patterns for different categories, at least in this data. So, the bottom line there is, oh, and here's kind of a plot for a different person's brain, but same data, same experiment, showing the discriminability. Here, orange, the dark color, the red, means the signal in that voxel is, this, if taken alone, is more discriminating than the yellow voxels, which, if taken alone, aren't very discriminating at all. So you can start to see information about what parts of the brain have a signal that allows distinguishing better than random between different categories of words. So the conclusion from this, of course, is we're thrilled. We're very happy that we haven't been wasting all of our time the past 6 months trying to train these classifiers because we found one that could be trained. I think this suggests some interesting possibilities. So for example, you could imagine trying, let's say, to build a brain dictionary for words. What if I showed a whole bunch of different words to people, looked at the response patterns for those different words, and then let's say, what if I clustered words according to how similarly the brain responds to them? It would be very interesting to compare the clusterings we get that way from, let's say, the clusterings we get out of statistical language processing on large corpus to see what kind of words get clustered, let's say, based on context. So we haven't done that experiment. I'm not even sure whether the data would allow it, would have a strong enough signal to do that experiment effectively, but this just kind of opens some possibilities that we don't quite know where the boundary line is between what's going to be doable and what's not going to be doable given the limits of the instrumentation. But it does mean that there's some hope. And I do want to finish, but I got to just show you one other experiment before I do. Let me come get at it after this, okay? So here's another experiment. In this case, this is what people in the fMRI machine saw. First they see a sentence, like that. Then they see a blank screen for 5 seconds. Then they see that. And then they have to answer true or false by pushing a mouse button. Half the time the sentence comes first, half the time the picture comes first, and the second comes second. Okay, so that's just another cognitive task. So we're interested in whether we could discriminate the mental state of, tell whether you're looking at the picture or the sentence that describes the picture. And in this case, again, we found that we could, with pretty good accuracy, 90% accuracy or so, on a subject-by-subject basis. And oh, I forgot to point out, the first experiment I mentioned was also subject-by-subject. So here's another example. In fact, there's the third one where we train the system to distinguish between sentences, whether you're looking at a sentence that you think is ambiguous or one that you think is unambiguous. And we could also, with about 90% accuracy, distinguish those two. So there are 3 examples that show that there is some potential to actually decode mental states of those particular kinds in these limited settings. We don't know where the limits of the technology would be. But there's one glaring limitation on what I just said here, which is that we have to train on individual subjects. And what would be great would be if you could train a cognitive state detector for, this person is confused by the sentence and have it work across people. Then that would be something you could publish on the web. Other researchers could use it as essentially a virtual sensor of cognitive state for their own work, if you could go there. So the problem with merging data from different subjects is that different brains look different. So here are brains from 2 different people. Again, what you're looking at is a 3-dimensional brain sliced up. And you see that they're really just different shapes, and the folds aren't in the same place. And so one interesting machine learning question is, could you come up with learning algorithms that would nevertheless be able to work across subjects? And in the last 2 months, Zhuri Wang, who is one of our students, got the first result, at least for us, of doing this, where what he did is he went back to he took a very simple approach, but it worked okay. he went back to that picture sentence experiment that I just described. And he said, well, the voxels, we can't really line up the voxels. The brains are different shape. The standard neurosurgeon methods of putting brains in the canonical shape are too poor spatial resolution. It won't work to do that. So what he did was he defined 7 anatomical regions in the brain, like Broca's region. Regions that are maybe an inch big or so, plus or minus, factor of 2. And defined anatomically in terms of where the physical structure of the brain is. And for those 7 regions, he just treated each of the 7 regions as a very big voxel and took the mean value of the activity within that region as the value. So now he's got a 7-dimensional instead of 30,000 dimensional time series image. And then he was able to train detector, whether you're looking at picture or a sentence, on a collection of subjects and test it on a different person's brain and get, again, much better than random accuracy doing it. So there's some evidence that might be, at least in this case, it is possible to train a classifier that works across brains by doing essentially physical abstraction. I think there are many more, that's my last slide. So to conclude, I think there are many more opportunities here for anybody who's interested in machine learning and signal processing, time series modeling and high dimensional spaces. Very little is known about how the brain works. A lot of very smart people have looked at fMRI, but have pretty much focused, because they had to, on low-level signal processing and how do we adjust for head motion while the person's in the machine and those kind of issues that are important. But now that they're kind of solved, we can start to build on top of those and do these kind of experiments. This is completely underexplored. The potential is obviously large, but also the uncertainty is large. We don't know what the machine will actually allow in terms of signal to noise and what will be detectable. So let me end there. And I hope that, if nothing else, you'll think of the next few years in AI as something that is likely to actually have much closer connection to fields that are interested in understanding natural intelligence. So, thank you.
So, on the first set of recognizing category words, so I have a lot of questions. Sorry? Oh, okay. So I have a lot of questions. So one is, you could say, well, for the artifacts, well, you don't perform well because maybe you don't have artifacts as kind of natural conceptions. If you look at kind of ways to, like, what categories are more natural now?
I have many of those same questions. We haven't even finished processing all the data that we have. I'm not sure whether this artifact is an artifact. I'm not sure whether the data I showed you there was from a single subject, and you don't see exactly that trend in every subject. There's one of the great uncertainties in this whole area is you don't know what's genuine individual variation and what's noise in your data from subject to subject. Although, of course, there are ways you could think of constructing experiments in a clever fashion to tease out those two. But the short answer to your question is I share the question and many others, and we just haven't gotten to most of them. I'll also tell you there are some things I don't like about this particular data set, and one reason that we haven't put our energy in there as much as looking at other data sets is we just finished collecting some new data with people looking at words that I think is much cleaner data and is going to allow us to make more defensible claims.
Yeah, I have one more.
One question at the back.
There was one over here.
question that I have for you is, I mean, when a person sits outside, let's say I'm sitting here and I'm looking at some words, I do have different reactions to the word when I'm sitting in a machine where I have
Yeah.
a screen. So, I mean, have you guys thought something about it?
Right, so let me tell you about the new data that we just collected. So this is data where, so in the data that I didn't tell you about the details of the data for that experiment, but in that experiment, people were presented words pretty much every 3 seconds in a block, and they were all words about food. And then there was some space and time, and then they were presented all words about buildings. So they're, it's called a block design in the fMRI world, but that's how the data was presented. In the new data, in the new that we just collected, we presented a word for 1 second, asked the person, is this a noun or a verb? And then the screen went blank for 14 seconds. And we spaced them out that much because we wanted to make sure that whole fMRI response decayed back down to baseline before they saw the next word. So in phase 1 of the experiment, they see these individual words. They're just asked, is it a noun or a verb? By the way, there are a dozen voxels that predict perfectly whether they said noun or verb, but that's a different story. Whether that's statistically significant is also an interesting question. Anyway, that's part 1. Part 2, we presented words, the same set of words, but 3 seconds apart. So we can see whether we can deconvolve and tease apart the response to different words. In part 3, we presented the same words, but we told people these are 2-word sentences. You have to answer true or false. But they were the same words. So, you know, like elephants fly, false. So now we have data from the same person in the same 20-minute period where they're looking at words and thinking of them as isolated words, and they just have to answer true, noun or verb. And then they're seeing the same pair of words in, coincidentally, the same sequence, but being told it's a sentence that they have to answer true or false. So we want to see if there's any systematic difference you can see in when they think of this pair of words as isolated versus a sentence. So I like your question, and we're trying to get... You can see that part of the game here is to be creative about how you design experiments, just to collect the right distinctions.
Also, when you had like plus divided by star, and you gave the next question whether it was above or below. Do you think like people are trying to remember the question and they are actually looking at star in that thing, or how do you guys model that part?
We don't model that part. So, well, we did not model that part. All we did was train a classifier to tell which of the two things you're doing, because we were interested in the question of whether in a single event we could detect which of the two. But we don't have a cognitive model of how people actually process that stuff. Nobody has posited one that they believed enough in that they were willing to do the hard work to try to fit it to the data.
I hope that fMRI isn't the last best kind of brain imaging that we have, because it has lousy temporal resolution, lousy space resolution, and it's very uncomfortable, and you have to do it in very expensive equipment. It's far away from where you would otherwise be. So what else is on the horizon that might either supplement or complement?
That's good.
Or complement these data?
That's good. So one is ERP, evoked response potential, where essentially you put just little electrodes on your skin and look at electrical activity. The bad thing about ERP is that it's even horrible spatial resolution. The good news is the temporal resolution is magnificent. So you can get down to a few milliseconds. One thing we actually have now is data where we have the same subject who solved mental algebra problems in the fMRI machine, then solved mental algebra problems with ERP sensors on them. Why didn't we do that at the same time instead of 2 different sessions? You can't have anything magnetic when you're in the fMRI machine. It's a very bad idea. So there's some logistic issues of doing simultaneously. There's another technology that somebody's telling me about just last week called near infrared, where this sounds spooky to me, but apparently you shine an infrared laser through somebody's skull and look at the backscatter. And it collects signal as far in as 2 centimeters. So that is also measuring blood flow fluctuation, so it'll have some of the same temporal problems that fMRI has, but it will not have the same cost. The fMRI machines cost several $1 million right now. So those are 2 of them. There are also things, I think something that's going to happen in the next 5 years is this. There are already, for clinical, we were talking about this at lunch, but there are already for clinical uses, various brain implants that people get. So there are cochlear implants for people who can't hear, and more recently the FDA approved retinal implants, and UCLA has implanted some of the first of those. So there are implants that generate signals directly into your nervous system. Well, once you've got those in, you no longer have there's no increased risk to putting sensors on them as well as signal generators. And so, in fact, you may be able to build a better cochlear transplant if you actually did some recording of signals to see what was going on. So I would guess over the next 5 years, some of those implants that are going into people for clinical purposes will begin to be used to collect data as well. On the more fanciful side, I was at a talk yesterday by Ray Kurzweil, who assured me that within 10 years, is it 10 years or 20? With Ray, it's all in the exponent in the log anyway. But Ray assured me that within some number of years during our lifetimes, there will be nanotechnology for putting sensors in through your bloodstream that will observe brain activity. He was a little fuzzy on the details. But he said that might be some number of years off.
On that note, let's thank our speakers. You can stay back.